to my wonderful PU2 champions. So finally we have a biology preparatory examination, and I know there is not too much time, which is why I'll not take away any bit of your seconds out there, my dear children. Just one very important thing, this video will help you with all the most important chapters out there. There are certain other chapters which I'm not covering because I know it will be overburden for you if at all I come up with everything all together. But yes, you have the PDF. Please refer whenever you go through bus or anything like that. You can just refer to the answers which are given in this particular PDF. Wishing you all the best, and the speed might be way too much. If at all you want to lessen the speed, go to the settings and lessen it up. Wishing you all the best, lots and lots of love. Let's go ahead. Going ahead with the first chapter that we have, and that is sexual reproduction in flowering plants. And the first is the very important question: what is the significance of sporopollen? Sporopollen is a very important and the most resistant organic material out there. It forms the exine layer of the pollen grain, and my dear children, it can resist alkali, acids, any pH, which is why it can be stored as pollen grain can be stored as what? Fossils as well. Always remember, this is part of exine. Inside that you have intine. Intine is made up of Priyanka Chopra, P for pectin, C for cellulose. Inside that you have plasma membrane, and inside that you have two very important cells. One is the very big cell, which is known as vegetative cell, and inside that floating, what do you have? You have the generative cell. Vegetative cell has an irregular nucleus. Generative cell has a spindle shape or elongated nucleus. Elongated nucleus would be more right, so write that. Now the generative cell can divide into two to form two male gametes out there, and so in that particular case, the pollen grain will be having 3 cells, otherwise it just have 2 cells. So 60% of the angiosperms, that is majority of the angiosperms, release the pollen grains in the 2-cell stage. 40% of them which are left are going to release the pollen grains in 3-cell stage. I hope that is crystal clear. Now going ahead, micropyle remains a small pore in the seed coat of the of the seed. Why so? So basically, if at all you see the structure of the ovule, my dear children, you have 2 integuments, and this is your ovule, and here you have the micropyle region. Why do you think it remains like that? Why do you think it remains like a bore? Because you need to make sure that once the ovule transforms into a seed, it gets enough amount of moisture, air and everything out there, which is why the micropyle is still there. Now apple and cashew are not called true fruits. Why are they not called true fruits? Because usually what happens is that this ovary which is there outside the ovule, it is the one that transforms into fruit. But in some plants, okay, some other parts of the flowers also contribute to what? Fruit formation, and that is known as false fruit. Apple and cashew are examples of what? False fruits. So micropyle remains as a small pore in the seed coat of the seed. Why? Because, my dear children, it helps with the entry of water and oxygen during what? Seed germination, which is very, very important. Apple and cashew are not called true fruits. Why? True fruits develops only from ovary after fertilization, but over here they are developing from other floral parts also. So if at all you see the structure of this particular apple, here you have mesocarp, endocarp and everything, which is the part of the ovary. So ovary is definitely taking a participation in formation of fruit, but the majority of the part is from thalamus. Thalamus is another floral part which is helping in the formation of fruit over here, which is why you call this as false fruit. Always remember that. Now differentiate between pericarp and perisperm. So the walls of the ovary that we have is known as pericarp. How many layers do you have? You have endocarp, mesocarp and exocarp. Exocarp is the outermost layer that we peel off. Mesocarp is the thing that we eat, and basically surrounding your seed you also have one more layer, the thick layer that we have, it is known as endocarp. Always remember. Now whereas when I talk about perisperm, what is perisperm? So my dear children, if at all you have seen the structure of ovule, okay, in the structure of ovule you have cells around over here, you call it as nucellus. Okay, initially that nucellus is there, later on it usually disintegrates or basically goes away. But over here in some, basically, wait just a second, okay, in some of the plants out there, this nucellus still persists. This nucellus still persists, and when in the ovule the embryo is being formed, the nucellus is going to provide nourishment to the embryo. Along with the endosperm and everything out there, this nucellus also helps in the nourishment of the embryo. That is known as perisperm. Only in 2 plants, okay, B square, that is beet as well as black pepper, you will be finding perisperm. So pericarp is the outer layer of the ovary, perisperm is something else. Okay, going ahead, difference between albuminous and non-albuminous seeds. Albuminous seeds basically means what? The one where the endosperm is still persistent. Listen, after double fertilization, 2 things happen. One is basically you get the zygote. The second thing is you get the endosperm. The endosperm is going to nourish the zygote to form the embryo. Sometimes the endosperm is used up in plants. Okay, it's used up in plants. How? Because when the zygote turns into an embryo, the embryo says that I can store my own food in the form of cotyledons. I do not need endosperm anymore. So the endosperm, whenever it is used used up. Okay, it is known as non-albuminous seeds, and here you will be finding embryo with 2 cotyledon out there. So whenever you can break the seed into 2 parts, just understand they are dicots. Example, bean, pea, groundnut, all of them I can break into 2. That means they have cotyledons instead of what? Endosperm. So they are considered to be non-albuminous seeds who have completely used up their endosperm during the embryo development. On the other hand, there are some plants which still have some endosperm left. Rest of the endosperm is utilized for formation of only 1 cotyledon. You call them as monocotyledons. You also say that they have what? Albuminous seeds, because some amount of endosperm is still persisting. For example, the things that you cannot break into 2, for example, wheat, maize, barley, castor, coconut, everything. Coconut you can break into 2 like this, but if at all you see the embryo, my dear children, you cannot break into 2. Okay, so this is albuminous and non-albuminous seeds. Now draw a labeled structure of two-celled pollen grains. Please remember, you have to draw everything which is over here. Super important. Okay, I have explained the structure of pollen grains. Now, LS of an embryo of a grass, this is also super important. Always remember, you have the plumule over here. Covering the plumule, you have the coleoptile. Before that, radical, root cap, and then here you have the coleorhiza. One scutellum is there, and that has been mentioned over here. The other part is epiblast. If at all you are asked about the dicot embryo, please do not worry. Just make the diagram like this. This is going to be your plumule. This is going to be your radical. This is going to be your root cap. And now from here, you need one cotyledon, which goes like this, and another cotyledon that goes like this. And your picture is complete. This region below is known as hypocotyl. This region is known as epicotyl. At the tip of it, you have plumule. Remember, over here, covering the plumule, you have coleoptile. Covering the root cap, you have what? Coleorhiza. So that is the only difference. Coleorhiza has an R, radical also has an R, coleoptile has a P, plumule also have a P. Over here, the cotyledon's name is also given since it is only one. It is known as scutellum. It is known as scutellum. Always remember, this is the embryo of a grass, and grass is a monocot. Grass is what? A monocot. Going ahead with the next thing, draw a neatly labeled diagram of typical anatropous ovule as an ulta ovule. So basically in ovule, what is there? You have 2 integuments like this, and then my dear children, it's inside the ovary. Inside the ovary, you have the placenta. Placenta is attached to the ovule with the help of a structure that is known as funicle, and the point of attachment is known as hilum. Now suppose this whole thing is shifted this side. Then you have the placenta over there, funicle over here, and hilum over here. This area where the integuments are not there, you call it as micropylar region, and this area where the opposite of micropyle, wherever it is known as chalaza. It is known as chalazal end, my dear children. Now the funicle can be here, still the chalazal end will be here itself because the micropyle will be here. So the opposite end is only considered to be what? Chalazal end. I hope that's clear. Anatropous ovule means ulta ovule, where this particular part is given over here, and I gave you the picture of this. Inside the ovule, you have the nucellus also, the cells of the nucellus. One of them is going to become the megaspore mother cell and going to undergo the meiotic division to undergo megasporogenesis. Only one functional megaspore is there, and this functional megaspore is going to form the entire embryo sac with the help of 3 mitotic division. Now, why are some seeds referred to as apomictic seeds, and mention its advantages to a farmer who uses it? So why are some seeds known as apomictic seeds, my dear children? So usually what happens, you have the female gamete, and the female gamete fuses with the male gamete to form a diploid zygote. But over here, the gamete is already diploid in nature. So basically, this particular seed will only able to, this particular plant which is there, it already has a diploid female gamete and it will form the zygote. So you do not need fertilization to happen. So now the plants which are going to be formed will be looking exactly like this female parent plant out there. And suppose this female parent plant is super good, it is pest resistant, this and that, every feature of it will be transferred to the seedling or the new plant out there. This is also termed as asexual mode of reproduction. It is usually seen in grasses and Asteraceae. It is seen in grasses and Asteraceae. It is genetically identical to the parent plant, which is exactly why farmers use them. Why? Suppose a farmer is having a good plant and now it wants to create clones of this particular plant, then in that particular case the hybrid nature is being maintained, which is why you can choose the apomictic seeds. Also, it is cost effective. You do not need to buy every time hybrid seeds out there. One plant is enough. If at all it is undergoing apomixis and producing apomictic seeds, you do not need to buy hybrid seeds every single time. And uniformity in the groups. All the crops, my dear children, are genetically going to be uniform as well. These are the reasons. Please go through them as well. Now, example, always remember grasses and Asteraceae. One more thing, a polyembryony is also given. Polyembryony means same thing. Over here, if at all you are forming multiple embryos within the, usually only one embryo is being formed, but think that multiple embryos are being formed. Here also fertilization is not required. Suppose the nucellus is still persisting. I told you in some cases persist, right? Some cells of the nucellus gets inside and forms embryo. In this case, you call it as polyembryony. You see this in citrus fruits. Citrus fruits as in orange, orange and this thing. What do you call? Mango, yeah, orange, mango and things like that. Now, plan an experiment and prepare a flowchart of steps that you would follow to ensure the seeds are farmed only with the desired set of pollen grains. So basically, if at all there's a crop improvement program and you want to produce hybrid variety of seeds, what do? You do hybrid variety of plants out there. What do? You do artificial hybridization, which looks like this, my dear children. So suppose a plant has two things, that is the male and the female part. You do not need the male part, otherwise the pollen grains will land on the stigma. So first thing that you do is emasculation. You remove the male part, emasculation. You remove the male part. Now what do? Basically you cover this particular plant up so that no other pollen grains can come and line until it becomes receptive. Now the moment it becomes receptive, what we do? The pollen grains that we want, we will put it over this and we will rebag it. We will do what? We will rebag it. Okay, there is bagging and rebagging also, and then we will wait for the entire process to happen. So over here the entire process is given down. I'm not reading out everything because the time is not up there. Okay, but yes, you were there in the live. If at all you were not there in the live, please download the PDF. You can read through if at all you're going through bus and things like that. Okay, but don't worry. And also one more thing, if at all the flower is not bisexual, that means it does not have male and female parts, then you need not do emasculation. Okay, you need not do emasculation. Now, importance of such experiment, they prevent contamination by unwanted pollen and help in producing hybrids with desired character. Now, this question can be asked in different ways, that a farmer wants to create a hybrid variety of plants. What crop improvement program will he use? Crop improvement program comes, then understand it is artificial hybridization that is being asked about. Now, picture related to pollination is given, that means insect pollination is given over here. You have to talk about, will this pollination confirm fertilization? Do you think pollination confirms fertilization? No. Pollination means landing on the stigma. Fertilization means fusion of male and female gamete. So pollen grains can land on the stigma. They can either also form the pollen tube, but not all pollen tubes comes and reach there. And sometimes they are not. The pollen tube, the pollen interaction, the pollen pistil interaction also does not take place, which is why fertilization need not happen, my dear children. Does the plant give some floral rewards to the insects? Yes, it gives nectar, it needs pollen grains, it also provides what? Shelter as well. Now, characteristics of flowers in insect pollination. Flowers basically are large, colorful and fragrant. They are sticky or spiny pollen grains so that they can stick on the surface of the insect, and the stigma is sticky to capture the pollen grains brought by the insect. The foul-smelling flowers are there in plants, pollinated by flies and beetles. Similarly, remember the examples of wind and water pollinated plant. Wind pollinated, you have grasses and corncob. Water pollinated, you have Vallisneria as well as this thing, Zostera. Remember, water lily, water lotus, even though they are in water, they do not undergo water pollination. Okay, now complete the given tabular column with respect to the male and female gametophyte of angiosperms for the mentioned character. Number of cells in the male gametophyte. What is the male gametophyte? It is the pollen grain. The one that forms the male gamete is the male gametophyte. What is the female gametophyte? It is the embryo sac. So my dear children, the number of cells present in the male gametophyte is what? Either 2 or 3. Okay, either 2 or 3. If at all you see the structure. If at all you see the structure. Okay, I'll just say. So this is vegetative cell, and then generative cell. The generative cell can divide into 2 also. So 2 or 3. If at all you talk about the female gamete, over here you have 3 antipodal cells, you have 2 synergid cells, 1 egg cell, 3 of them together comes and forms egg apparatus. And then you have 1 big cell which is known as central cell. The central cell has 2 polar nuclei. So 1, 2, 3, 4, 5, 6 and 7 cells is there, but nucleus, how many is there? 1 extra nucleus is there. So you call 8 nucleate 7 celled condition. Okay, a 7 celled 8 nucleate condition up over here. Always remember that. Now if at all you solve this, okay, just a second. Now if at all you try to solve this, so you get number of cells present in the male gametophyte 2, 1 vegetative, 1 generative, or 3, 1 vegetative and 2 male gametes. Over here you have 7 cells. The name of the 7 cells have been mentioned. Number of gametes present in, there are 2 gametes present in the male gametophyte, whereas there is only 1 in the case of mature female gametophyte. Type of cell division involved in the development of the male gametophyte. To form the male gametophyte, you need to undergo mitosis. In the case of embryo sac also, you need to undergo mitosis. So over here, the microspore from the microspore which was there, that forms the male gametophyte. When it forms the male gametophyte and undergoes mitosis to form the 2 cells out there, then again mitosis to form the gametes out there. Similarly, here 3 rounds of mitosis happens in the case of female gametophyte. Please remember it. Now, the ploidy of cells, they are all haploid. They are all going to be haploid in the case of male gametophyte. In the case of female gametophyte, my dear children, not all cells are haploid. Why? This haploid. But central cell, it has 2 nuclei. 1 nuclei haploid, another nuclei haploid, diploid in nature. Since it has 2 nuclei, central cell is going to be diploid in nature already. Okay? So please remember that as well. Now, what is there? Advantages of seed. Seed provides what? Protection and dormancy. Protection and dormancy, my dear, because seed coat is there. Remember the integuments which are there in the ovule, they transform into what? Seed coat, testa and tegmen, and they provide protection and also dormancy. Food for the embryo. The seed has endosperm, my dear children, which is why endosperm or cotyledon, which is why both of them are going to provide nutrition. Dispersal also. Seeds are super important for dispersing by wind, water and everything out there. Cool? Cool. Now, next thing, explain the different outbreeding devices by the flowering plants. So, one is outbreeding means you do not want to undergo pollination, you want to undergo cross pollination. Those are outbreeding devices. So, first thing is time difference. Pollen release and stigma receptivity is not synchronized. Second, spatial difference. That means anther is over here, stigma is over here. Pollination will not be happening. Self incompatibility. This is a genetic mechanism. But even the pollen grains land, the pollen postal interaction will not be taking place properly. Now, the next one is unisexual flowers. Now, there are some plants which are unisexual. Plants, as in one female plant, one male plant. In that, obviously, what is going to happen? Cross pollination is going to happen because only female plants are over here and male plants are over here. That is dioecious plants. Example is papaya. You call them as dioecious, where the sexes are separate in two different plants. For example, papaya, female plant is there, male plant will also be there. There are monoecious plants such as castor and mace also, where similar things happen, where male and female flowers are there in the same plant, but male and female flowers separately. So over here also, to an extent, cross pollination is going to happen, but geitonogamy can also happen. Geitonogamy can also happen up over here. Pollen from the one flower to the another flower of the same plant. That can also happen. Okay, so please remember that. So it is not true cross pollination, but yes, to some extent it can prevent it. Okay, double fertilization is a unique event arranged. Okay, double fertilization is a unique event. I hope you know that one particular thing which is there. Okay, one particular male gamete fuses with the female gamete to form the zygote. The other male gamete fuses with the two polar nuclei. So male gamete has a nuclei. These two nuclei are also there. So there are three nucleus which are fusing, which is why you call it triple fusion. And the end product is endosperm, where you have triploid nature. Zygote is 2n and that is 3n. So this is known as syngamy. Okay, this fusion is known as syngamy and that is known as triple fusion. I hope that is clear. And now we go to the next chapter, that is human reproduction. My dear children, obviously there might be some other topics as well, but this video intends to just help you with the last minute revision at least, because obviously at last minute you cannot revise everything, right? So this is it. Going ahead with the other things, that is human reproduction. The first thing is what is menstrual cycle. Mention one pituitary and one ovarian cycle, ovarian thing. So see, this menstrual cycle questions would be very easy for you if at all you look at this picture very carefully. So from the 1st to 5th day, you have the menstrual cycle, menstrual phase. Okay, what phase? Menstrual phase. In menstrual phase, the level of estrogen and progesterone is going down. The level of estrogen and progesterone goes down. And when these two guys goes down, my dear children, the pituitary hormones, you have two pituitary hormones and two ovarian hormones. These are the ovarian hormones, these are the pituitary hormones. FSH and LH goes up. FSH and LH slowly goes up, and my dear children, in this particular period, slowly after that, the follicles are going to develop. Follicles are going to develop. Now, when these follicles are developing under the influence of FSH and LH, one very important thing happens. The granulosa cells of these follicles are going to release some estrogen, which is why the level of estrogen is going to up. Now, menses happened. Now, slowly endometrium is going to regenerate under the influence of what? Estrogen. So, which is why you call it proliferatory phase, because this thing is proliferating, and you also call it follicular phase because follicles are also developing. Now, what happens is that when the level of estrogen goes really high, it causes LH and FSH surge, especially LH surge. At this particular point of time, the ovulation happens. The mature follicle breaks open and releases the secondary oocyte. This is known as ovulation. This happens in the mid of the cycle. Suppose the cycle is of 28 days. On 14th day, you will be having ovulation. You will be having ovulation. Okay. Now, my dear children, whatever is left behind in the follicle, you call this as corpus luteum. And this corpus luteum is staying because LH and FSH is there, especially LH. Now, corpus luteum, my dear children, will be releasing some hormone, that is progesterone, and also estrogen, which is why the level of progesterone goes really high. If the level of progesterone goes high, my dear children, endometrium also thickens very much, because this is a very important hormone out there. And also estrogen is also there. This phase is known as secretory phase, because now the endometrium which is there, it is starting to secrete some very important things, thinking that embryo will be coming and basically I have to nourish it. But if at all that happens, it happens, otherwise not. So it is also known as luteal phase. Why? Because corpus luteum is doing everything out there. Now, what usually happens is that progesterone, when its level is very much, it does not allow FSH and LH to go up. It makes it drop. And when they drop, my dear children, corpus luteum will not be maintained, which is why the progesterone will also go down and estrogen will also go down, and again you have the menstrual cycle happening up there. Now the question is, what are the functions of the hormones in the menstrual cycle? FSH is a pituitary hormone. It stimulates the growth and maturation of ovarian follicles and also induces the follicles to release what? Estrogen. Estrogen, what does it do? It basically repairs and proliferates the uterine endometrium and also leads to LH surge, causing what? Ovulation. These are the important things that you have to keep in mind. Now, what is placenta? List its function out there. So, placenta is something that is being formed partially by the embryo and partially by the uterine tissue that is there, okay, mother's tissue out there. So in the embryo which is going to come and settle up over here and form the uterus, it is known as blastocyst embryo. Blastocyst stage of embryo, where the outside layer is known as trophoblast, and my dear children, the inside layer is known as inner cell mass. So when this comes and attaches on the endometrium, it forms finger-like projections like this. You call this as chorionic villi, and from the mother's side also you have some maternal tissue rounding up together. Now this forms a thing that is known as placenta. Now this inner cell mass which is there is going to form the actual embryo out there, okay, going to form the actual embryo out there. So placenta is a vascular connection between the mother and the fetus that helps in nutrition, respiration, excretion and hormone secretion. How does it do that? It's written over here. Respiration as in supplies oxygen, removes the carbon dioxide. Similarly, excretion removes all the metabolic waste out there. And also it's a temporary endocrine gland. Why? Because it releases certain very important hormones such as what? HCG, human chorionic gonadotropin, HPL, progesterone, estrogen. There are 4 hormones released by placenta. Please remember, only 4 of them are there. Okay, only 4 of them are there. During pregnancy, there is an increase of 3 other hormones also: cortisol, then what do you have? Cortisol, prolactin and thyroxine. Cortisol, prolactin, thyroxine, their level also increases in multiple folds in the blood during pregnancy. They are also already there, they increase their production. Okay, now ovary which is there, it also releases chapter that we have is human health and diseases. But as I said, we do not have time, so I will not go to entire thing. I'll just tell you what is important. So please remember, ringworm disease is caused by 3 types of fungi, that is trichophyton, epidermophyton and microsporum. Ascariasis is caused by what? Ascaris lumbricoides, which is a Ascalminthus, and that is a filariasis is caused by Wickeraria bancrofti or Wickeraria malai, and is spread through mosquito bite. Okay, please remember that. There are 3 ways in which you can detect cancer, that is biopsy, histopathology, CT scan, that is computer tomography. With the help of that, you will be able to find if at all there are some internal organs that have tumors or not. MRI is there to find defects in the living tissues as well. But anyway, if at all the question comes, what are the methods through which you can find cancers of internal organs? Write the 3 of them together. Now, what are the features of cancer cell? First, uncontrolled growth. Second one, they have loss of contact inhibition. They will not stop dividing. They will keep on dividing even though they come in contact with other cells. And also metastasis. They will go spread around throughout the body. Now, my dear children, the next thing that we have is the lymphoid organs. Remember, there are 2 primary lymphoid organs where the lymphoid cells are going to be formed. They're going to mature and they're going to be antigen sensitive. Okay, they're going to be sensitive to antigens. They don't start fighting up there. Where do they start the fight game? In the spleen, in the lymph node, in the Peyer's patch, in the malt. So that is secondary lymphoid organs where they become effector cells. So effector cells is different from what do you call antigen sensitive, being antigen sensitive. The next thing, very important thing, is the replication of retrovirus. Please go to the picture. They have RNA. They insert the RNA to our cell. In our cell, they use reverse transcriptase enzyme. Their enzyme to convert the RNA to DNA. And now this DNA gets incorporated into our cell, and this DNA, using our machinery, it forms viral proteins as well as viral enzymes and viral RNA and everything out there, and then the viral babies will be moving out. The first cell where it goes in effect is macrophages. It is known as HIV factory, but usually it affects the T helper lymphocytes. So T helper lymphocytes are the ones which are usually being affected. Also remember that the antibody structure, 2 heavy chains it has, 2 light chains it has. This is the antigen binding site. Also remember that. Okay, difference between innate and acquired immunity. Innate is there from us from our birth. For example, our skin, our mucus, everything. So cellular barriers, cytokine barriers, you have this thing, cellular, cytokine, physiological barriers coming under what? Innate immunity. It's present from birth. Acquired immunity you develop after an infection or vaccination. It's non-specific over here. That is very much specific to antigens. There is no memory. Here there will be memory. Always remember that if at all the question is a virus infected cell is releasing something to save the other cells from virus, it is interferons. They come under innate immunity itself. Okay, so please remember that also. Now, apart from that, ascariasis and all I told you. Okay, why do cancer patients get alpha interferons? What are alpha interferons? They are biological response modifiers. It increases the immunity of the people who are fighting with cancer. Always remember, this interferon is different. Now, name the group of genes that has been identified in normal cells that can lead to cancer. So, in a normal cell, we have proto-oncogenes. When they become activated, you get oncogenes, which basically causes cancer. Okay, so proto-oncogenes, they are problematic. So these are the very, very important things. Apart from that, if someone comes with snake bite, my dear children, snake bite, they are given antivenom. What does this antivenom contain? It contains ready-made antibodies. So this is a type of passive immunization. If ready-made antibodies are given, this is passive immunization. If at all your body is producing the antibodies by themselves, it is active immunity. For example, we are getting an infection, our body produces what? Antibodies. Whenever we get vaccination, we get antigens, weakened antigens, but our body produces antibodies. So vaccination is a form of active immunity. Whereas if at all we are given tetanus injection, or if at all we are given antivenom, that is what you call passive immunization. From mother to child, ready-made antibodies are given, IgA, in the form of milk, through the milk, right? A for Amma, you can remember like that. So there is ready-made antibodies. From the mother, from the placenta to the baby, you get IgG, that is again ready-made antibodies that is given. During allergy, you have IgE, which is increased. Allergy, IgE, please remember that. In allergies, you have histamine production, which causes the overall problem. Histamine leads to overall issues, and to treat it, you have antihistamines, you have adrenaline, and also you have steroids. So these are the few things. Obviously, there are more to it. My dear children, I'll request you to download the PDF that you have been given in the channel, WhatsApp channel. Go through it whenever you have time. I know you might be in bus or somewhere. Just read through the questions out there. It is definitely going to help you out. This is what I could do. I hope this could help you, and I hope the exam goes really, really well, because you guys are awesome, and I am pretty sure you will be doing great. Wishing you all the best, my dear children, and take care of yourselves. Do not be worried. Do not be panicked. Okay? Bye-bye. Take care.